Okay, for this first video, we're going to be talking about the cell cycle. So when the cell goes through its life cycle and it needs to replicate or use the process of mitosis or meiosis, it must go through a cycle. So the cell cycle has a life cycle like ours. It starts out in G1 phase, then it goes to the S phase, G2 phase, and finally the M phase. So in the G1 phase, the cell grows and develops. In the S phase, the DNA is replicated. In the G2 phase, the cell continues to grow, but there are also proteins that form to help in the replication process. So proteins form for replication. And then in the M phase is called the mitotic phase, and that's where the actual splitting apart of the cell and replication happens. And there's 5 different parts of the M phase. It's called prophase, prometaphase, metaphase, anaphase, and telophase. So we're going to talk about that now. So once it goes through its life cycle and gets to the M phase, it begins to split apart. So I'm going to draw a cell right here. So in prophase, the cell begins to break down its nuclear envelope. So here is the cell. Here's the nucleus with all of the DNA in it. So in prophase, the nuclear envelope breaks down. Begins to break down. Okay, here we go, prophase. So, here we go, it's beginning to break down. Then we go to prometaphase. In prometaphase, the rest of the nuclear envelope breaks down. And spindle fibers form and attach to the DNA. Okay, so right here we're in prophase, prometaphase. There we go. Envelope's gone. These spindle fibers are formed and attached to the DNA, like that. Then we get to move on to metaphase. In metaphase, the DNA lines up at the center of the cell. Okay, now we're moving on. Right now we're in prometaphase. So now during metaphase, the spindle fibers allow the DNA to line up here at the middle of the cell. They're still attached to the DNA. Okay, now in anaphase, this is the step after metaphase. Moving on to anaphase. In anaphase, the spindle fibers pull the DNA apart. So when it pulls the DNA apart, they go to opposite ends of the cell. So right now this is metaphase. The DNA is lined up in the middle. So during anaphase, the spindle fibers pull the DNA apart and they go to opposite ends of the cell. And they're still attached by the spindle fibers. Keep that in mind. Okay, now what happens is telophase. Telophase, the spindle fibers take themselves off of the DNA, removal of spindle fibers, and the nuclear envelope reforms, and a split in the cell starts to happen, which is for the next step. So cell has, it's called a cleavage furrow. Okay, so now, still in anaphase, what happens is the spindle fibers take themselves off of the DNA. The nucleus, the nuclear envelope, reforms around the DNA, and the cell prepares itself to split in half by making a cleavage furrow. So what a cleavage furrow looks like is it looks something like that. So now, the final step, which isn't part of the original 5 phases, is called cytokinesis, and I'm going to write this in a different color. In cytokinesis, the cytoplasm splits. So you have to have this cleavage furrow in telophase so that you can lead into cytokinesis, where the cytoplasm will split. So cytokinesis, telophase, the cleavage furrow is broken up, and we get a full split of the cell. And now we have 2 different cells. So throughout this process, checkpoints are created throughout the process to ensure that nothing goes wrong when the cell splits. So I'm going to write down the different things that we've already talked about. We've talked about G1, S, G2, M. These are all the phases. Then we've talked about prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis. Now that's a lot of steps for the cell to go through in order to split. So there's checkpoints that are created in this process so that everything goes smoothly. So the first checkpoint is called the G1S checkpoint. And this checkpoint is basically just ensuring that the cell is ready to replicate. Because remember, in the S phase, that's when the synthesis starts to happen in the DNA to where it's able to split once it gets to the M phase. So it's just making sure it is ready to go. The next checkpoint is called the SG2 checkpoint. This is just a checkpoint ensuring that the DNA replicated and has synthesized properly, so the cell can move on to prophase. So it's just checking the DNA. The next checkpoint is the G2M checkpoint. And this is ensuring that it's ready to move on to prophase. And then finally, the metaphase and anaphase checkpoint. I'm just going to do meta, ana checkpoint. Is ensuring that the cell, that the DNA splits completely from each other. So DNA moves to opposite ends completely. And that's the full cell cycle.